It's my pleasure, first of all, to say we're so pleased to be partnering with the College of ACES here on campus and the International Food Group and the International Office at ACES, who's helped sponsor this meeting. We have other sponsors as well, and we'll talk about them later, and supporters. So thank you again for being here. I think we're going to have a great program the next 2 mornings, and then a number of you will be here over the next 2 days in the afternoons as well, working on new collaborations around these topics that we put together. So we're here today for Machine Learning Farm to Table, and to get started, we've got a keynote. Dr. Schnable's here from Iowa State University, but it's my pleasure to bring up first the Director of the College of ACES Office of International Programs and Director of Food Security at Illinois, Dr. Alex Winter-Nelson.
Thank you, Melissa, and I mean that personally and institutionally. Thank you personally for your efforts to pull this together this morning and tomorrow, and institutionally for reaching out all the way south across Green to the College of ACES, which is the College of Agricultural, Consumer, and Environmental Sciences for those of you from farther away. I'm Alex Winter-Nelson. I'm the Director of the Office of International Programs in ACES, and it really is a pleasure to be here at this event. It was about a year ago that my office launched an initiative on big data, food security, and the environment under the leadership of Kathy Bayless and some other people I think who are in this room. And the vision that we had was to try to combine the agriculture and food sciences expertise at this campus with the computational sciences expertise that the campus is well known for, and to link that with private sector initiatives and other researchers around the country and the world who are working in this space that combines ag and data analytics. And the purpose of doing that would be to better address the global issues of food insecurity and environmental change. We at the College of ACES imagined that we would leverage new methods in data analytics, new data itself, new types of data collection that would allow us to better understand food systems from farm to fork, or actually beyond, sort of full cycle from soil to soil. And that with that understanding, we could, in fact, come to conclusions that would be useful for agriculture and food systems, both in this country and other developed countries, and in the developing world, an area where we identified that data was more scarce maybe than people realized, and that analysis of that data was more nascent than people realized. That kind of partnership of the ag sciences and the data sciences was going to require institutional partnerships through things like the Big Data Hub, NCSA, and others. And so I'm very pleased about this event happening. It represents progress, to my mind, in breaking out of silos and really finding collaborative relationships that can move us to another level. I regret that I cannot spend the full day here with you or be here tomorrow because of other travel commitments, but I take great satisfaction that you're here and that the event's happening. I also take great satisfaction in introducing the keynote speaker for this morning. The workshop organizers, I think, will confirm that it remains a challenge to get a group of people together that combine agricultural expertise, data sciences expertise, public higher education, and private sector. It's hard to pull a group like that together unless you can get it all in one person. And that's what they've managed to do with Pat Schnable, the keynote this morning. Pat's distinguished professor at Iowa State University. He holds an endowed chair and directs the Plant Sciences Institute that's fostering collaborations among plant scientists, engineers, and computational scientists. He's also the Changjiang Scholar Professor at China Agricultural University. Pat Schnable received his BS in agronomy from Cornell and a PhD in plant breeding and cytogenetics from Iowa State University. He conducted postdoctoral research in molecular genetics at the Max Planck Institute in Köln, Germany. His investigations of the maize genome have resulted in over 120 peer-reviewed publications. He's a fellow in the American Association for the Advancement of Science. He serves as consulting editor for PLOS Genetics, and he is past chair of the American Society of Plant Biology's Science Policy Committee. He's also past chair and a current member of the Maize Genetics Executive Committee, and he's co-lead of the Genomes Fields Initiative. If that's not enough, moving towards the private sector, Schnable is co-founder and managing partner of Data2 Bio, a limited liability corporation that provides research support for the breeding industry. Similarly, co-founder and partner of Dryland Genetics, LLC, which is developing crops for arid environments via next-generation breeding, and of Decision PX, another limited liability corporation that generates image-based decisions for crop production and breeding. He also serves on the scientific and advisory boards for several other companies. So I'm very pleased if you would join me in welcoming our keynote speaker, Professor Pat Schnable. And I really didn't make the lights go out.
Thank you very much.
But maybe you can make them go back on.
I don't know. We'll see. What's the prognosis here? While he works on that, I'll tell you a story about my graduate advisor, who was very good maize genetics. Talks were okay. And one time he was giving a talk in the department. This was in the era of slide carousel. Remember that? You put the slides in, and there was a ring that you would tighten on. I'm going to tell the story anyway. And in any event, he was using German slides which had thick glass, and they got stuck in the carousel. So one of my fellow graduate students went up and unscrewed the ring, de-stuck the slide, and things started again. The next one got stuck, and he took the carousel, my colleague, took the carousel off, turned it upside down to de-stick, and of course all the slides came out. So Peter Peterson said, "Don't worry, Brian. Just put them in random order, and I'll give a talk." And it was the best talk he ever gave. All right. I'm very happy to be here. I think this is absolutely the right thing we should all be doing, and I look forward to the next 2 days. So I'm going to talk about a bunch of stuff that tries to capture it. As I was sitting here, I realized, and this was not intentional, but you know, this is the diversity of maize upon which all of the advances in breeding have built. This is a production farm out in Iowa. I try to take my students and sometimes colleagues out to these for tours every few years because most people working in this area have never been on a production farm. This shows the controversies that go on with the adoption of new technologies, and this, although it's several years old, I think really encapsulates our vision at this meeting here. So this slide is a good news, bad news slide. And I will start with good news, and then we will quickly transition to the bad news and the challenges. So in the upper left here is a plot time on the x-axis, corn grain yields in the U.S. over time. And what we can see is up to the mid-1930s, yields were essentially flat. And then they started rising in an almost linear fashion. What happened in the mid-1930s? Somebody. Speak up. What? Fertilizer. Fertilizer, more generally? Application of technologies. Fertilizer was part of it. The big one was introduction of hybrids at that point. But even more important, again relevant to this workshop, is this is when statisticians started helping with field plot design so that you could overcome, or control rather, for the variation in soils across fields. This is also a good news panel. These are grain elevators. So when farmers harvest their corn, they store it in these elevators. Behind here, you can't see it, are railroad tracks with railroad cars. So the corn is brought from the field into the elevators, loaded into the train cars, the railroad cars, then taken out to the Mississippi and put on barges and then shipped around the world. This was a very good year. This photograph a little west of Ames, and there was so much corn that there wasn't room in the elevators, so this is a pile of corn that was just deposited on the ground. And to give you a sense of scale, my wife and son. So this illustrates the extraordinarily fruitful, productive collaboration involving plant breeders, agronomists, and producers. We've been, as a group, as a society, tremendously successful at increasing yields and producing enough food. This plot, now we transition into the bad news and the challenges, is similarly the x-axis is time. The y-axis is private sector R&D investment in breeding. Inflation adjusted, that's important. So what this means is the cost we make linear gains, and the cost of conducting those linear gains is going up every year. So if you extrapolate, and we're now at $2 billion a year, this ended a while ago when the company stopped sharing their data with the USDA, it is not a sustainable system. We can't continue to spend more each year to make the same gains. So the only approach, and again, if we look underneath this red line that I overlaid on top of Troyer's graph here, you'll see there are a number of different regression lines. Each of these is the introduction of a new technology. So just as in the mid-1930s, hybrids and statistical plot design and agronomic practices led to increased yields, each of these different regressions is the introduction of new technologies. And that's really the focus of what I want to talk about today, and I think is really the focus of this two-day workshop, which is that we need to think about what are the technologies that will keep us moving the curve in the right direction and maintain cost at some reasonable R&D cost, because we simply can't continue to spend more every year. For this group, the challenges here in agriculture, increasing demands for the products of agriculture, and we have less land. This is review for most of you. The agricultural inputs become more expensive, have reduced availability, especially water's a big one. I remember a number of years ago, somebody who was quite wise told me that eventually there would be wars fought over water, and I didn't believe it at the time. It's quite clear that that will almost certainly happen. And that we are recognizing the undesirable ecological footprints of agriculture in ways that modify how we're going to constrain how we will conduct agricultural activities. But it's really this bottom, the last bullet, that is the one that I think should concern us the most, which is achieving yield stability in a world with increasingly variable weather patterns. And in that light, this book is something I can recommend. I have not completed it yet. I am savoring it. It's describing the effect of climate change on the southwest United States. And when you come home from one of those days where it's absolutely miserable and you feel terrible because everything has gone wrong, you can open this book and realize that your small problems are indeed small, and there are much, much worse things happening, and sometimes that can help you sleep. At least it works for me. But as I say, this is really the big one. I mean, we need to continue to produce more food because there's more demand for it, but if we don't have yield stability, you know, it doesn't matter if our average yields are good. If we have some years where we have catastrophic losses of yield, we will have a nightmare. And it's not those of us sitting in this room that will probably suffer, but it's the several billion people at the other end of the income scale globally. So this is really a big challenge, and it's not like we have a solution in our back pocket. Developing yield stability is going to be extremely challenging. But it was problems, challenges, such as we presented on the last slide, that led the National Science Foundation to invest $32 million a number of years ago to sequence the corn genome, which was published in 2009. And since that time, the amount of DNA sequence data for corn in public databases has soared. So we have lots of data about the genetic diversity of this species. And our challenge then is how to use these data and other data to continue to improve this crop and, importantly, to build in yield stability. So one of the approaches that we can use here, which is the one I'm going to talk about today, which is really to use the big data paradigm to build predictive models, to exploit massive amounts of genotypic data, which, as I've said, are very readily available, combine that with large collections of phenotyping data and environmental data to develop predictive models, such as this very simplistic one here, which says that the genotype, the environment, and importantly, the interaction of those two factors, G by E, is what controls the phenotype or traits such as yield. And just for anybody who's, anyway, crop management is included here in the environment. So how do we build these? Well, first let's talk about how these predictive models will be useful to us. If we can build models that are truly predictive, we will be able to increase the rate of genetic gain per year. This means we won't need to test all of the potential new varieties in the field. We can identify those that are probably the dogs, get them out of the testing pipeline, and focus our limited and expensive testing resources on those lines that are most likely to prove useful in the field. We should be able to breed crops to withstand the increasing weather variability, because if we understand the genotype by environment interaction, and can breed for it, we can build crops that actually continue to produce reasonable yields even when the weather goes the wrong direction. We should be able to improve our ability to provide farmers with evidence-based recommendations for which varieties and which management conditions to employ in a particular year, which should lead to increased yields and, even more importantly, increased yield stability. And then these predictive models will enable daily national and global yield predictions or production predictions, which should facilitate early responses to food emergencies around the world. This is a really important one. We need to know before famine strikes that it's likely that one is coming. These models will also allow us to avoid market failures. So in the developed world, farmers employ various financial tools that depend to fund their operation and to hedge risk. And these depend on functioning futures markets. And if information about what's happening out in the world becomes asymmetric, if some organizations have good predictions about what yields and hence prices are going to be 3 months from now, and other groups don't have that access to that information, markets will fail. For the same reason you wouldn't buy a car from me if you can't let your mechanic, if I won't let your mechanic look at it before I sell you this used car. Okay, so if we want to build those models, what do we need to do? Well, first of all, we need an enhanced understanding of how diverse factors, genotype, environment, and this interaction term, influence phenotype. We have to understand that model, that equation that I showed previously. And then we need lots of data. There are statements made that we're in a data-rich world, and that is true in some disciplines. In agriculture, we still need a lot more data. Genotypes are fairly straightforward, but there are some constraints there that we probably won't have time to talk about today, and the environment and management practices. There are efforts to gather these, but particularly the phenotypes, when we get beyond yield, there is not a great amount of data. We need to develop new phenotyping technologies to help us solve this shortage of phenotyping data, sensors, robots, unmanned aerial vehicles, and then we need new methods for analyzing the resulting data, because this is a huge bottleneck. And then we need a community of plant scientists, engineers, and computational scientists who work together on this. So I'm going to address each of these in turn. I'll start with these first 2 bullet points. So this enhanced understanding of the biology as well as data. So weather, as we know, varies from location to location within a year, and between years we also see enormous variation in weather. This is a particular drought monitor. And we know that this weather, this E term, affects the phenotype, but we also know that there's interaction between genotype and environment. So some genotypes respond to certain environmental conditions differently than others do, and that gives us this G by E term. And this is really one of the big challenges, is to understand this. In order to understand that, we need data. We need information about the genotype of lines. We can get the weather data or the environmental data, and we need the interaction term. I have a lot of respect for this business model of 23andMe. How many of you have had yourself genotyped by 23andMe? Just a few of you. I can recommend this as a fabulous way to start discussions within a family, is to offer genotyping services to your family members, the ones that don't work with genotypes every day, and it stimulates some really exciting, interesting discussions. So her model is, she's built a genotyping chip. So all that's required is a little bit of saliva, I think it is at this point, but it's a cheek swipe. Maybe there's some cells in there. And customers pay to have themselves genotyped. If you do it on sale, it's about $99. But just like Kodak used to give away the cameras, essentially, in order to sell film, which some of you won't remember, but anyway, it's a model. It's a very interesting business model. She's not making a lot of money on the genotyping. What she wanted to do was, in fact, to get customers, and they've got about 1 million of them now, and 80% of those customers agree to share health data and lifestyle choices with 23andMe. That gives her a very valuable resource, which is a collection of individuals, about 800,000 people, for which she has the genotype, and then she sends surveys to them, and they talk about how much exercise they get, how much they eat, what they eat, family members' medical histories, their own medical histories, and she and her team can start making associations between variation in the human genotype and health outcomes. And those associations are enormously valuable, which she then licenses to the pharma industry. Fabulous business model. I have struggled for years to convince corn plants to pay me to genotype them and then to ask them to email me their phenotypes. Well, we haven't succeeded with that, but instead, a group of us in the maize genetics community established this Genomes to Fields initiative, and within that, there's this multi-state Genotype-by-environment project. It's designed to collect data of public lines that have been genotyped, grown at all of these locations around the US, and phenotyping data are collected using standard operating procedures so that it's very comparable. The data are comparable from all these sites. And we are also collecting, of course, environmental data for all those sites. So we are collecting data sets that will allow us to answer this challenge that I've set out for us today. But while we wait for those data to accumulate, because we really need multiple years of data, we've been mining historical data. So since the 1930s, experiment stations have been conducting yield tests at many locations around the United States. So these are all locations where various experiment stations have been conducting yield tests. And those data reside in a repository. Again, some of you will recognize this. Some of you may never have been in one of these. But in the stacks, there are books. And if you open those books up and you have the right software, you can look inside there and you can actually see there's a variety. And we have the number of ears per plant, and we have the ear weight and the percent dropped ears and yield, and various other phenotypes have been collected. Now, the challenge is that every state organized the data differently. Every state, as far as we can tell, has changed the way they organize and present the data over the years. The quality of the printing presses varied enormously, so it's actually rather difficult to convert these images into machine-readable format, but we've made some progress. So for Iowa, we have 94 years of data. We were able to collect all of the hard copies, and all of that has now been converted to machine-readable. Kansas, we have 76 years of data, and again, it's all machine-readable. Illinois, we're working on it. We've got hard copies now of everything back to 1934, but we're in the process of converting that. Wisconsin, we only have 42 years, and we're missing 4 years in there, but we've made some progress. So once we have these data, they are useful for trying to understand a number of things. But remember, we don't have genotypes, or maybe not remember, but we don't have genotypes for these lines. Most of these lines no longer exist. I would say almost all of them no longer exist. And even if we did, naming conventions have changed. It'd be very hard to associate genotype with those lines. But we do have the name of the variety and the company that produced it. And of course, we know where the experiments were conducted, and we have the phenotyping. So Dan Nettleton, who is an extraordinary statistician at Iowa State, has been using, in collaboration with some of his colleagues as well as graduate students, has been using these historical data to predict yield. So they've asked if they use a subset of the Iowa data as a training set, can they predict yields of varieties that have not been tested in the training set in the future, and they are achieving very reasonable accuracies. Dermot Hayes is an ag economist at Iowa State, and he's very concerned about this asymmetric knowledge about prediction, asymmetric access to accurate predictions about production, which drive costs. So Dermot Hayes is an interesting guy. When corn was first being converted to ethanol, he realized that once you start converting grain to ethanol, you have fixed the price of Iowa farmland to the price of petroleum. And unlike some economists who would simply write a paper about that, he went out and bought 3 farms because he realized what was going to happen. And so he's treated as sort of a god in his department because he bet his family savings on this principle, which turned out to be quite a good decision. So he's worried, though, about this asymmetric access to predictions. So working with Shamek and Bhaskar, who's here, they're using artificial intelligence approaches to mine historical data to come up with predictions of production in future years. We're also working with Satirius and Mike Castellano, who are using process-based crop growth models. So they're not using very much historical data. They're collecting very detailed measurements of crops, soils, and weather from fields in Iowa and using that to try to make predictions. But they're on this slide because this group is beginning to talk together, and my hope is that they will coalesce into finding best practices and synergies among these different methods. But we are limited in the understanding of the physical world. There are a lot of data that are available out there, weather and rainfall, temperature, wind speeds, relative humidity, and so forth. Liang Dong at Iowa State is an engineer who builds sensors, and he built an in-planta nitrate sensor. So nitrate is a very important nutrient in well, crop growth, plant growth generally, particularly for corn. But he's developed these sensors. These are about the size of a small notebook, and each one is able to measure nitrate concentrations in the stalk of a corn plant in real time. You have a probe that actually enters into the stalk of the corn plant. So this will allow ultimately, first researchers, but ultimately growers, to actually recognize how much nitrate is moving through the plant and adjust applications of fertilizer to maximize whatever you're trying to maximize, whether it's yield or economic profit or minimize environmental impacts of agriculture. I want to talk about another case of mining historical data, but first I want to develop this concept of genotype-by-environment interactions a little bit more. So if we have historical data from multiple locations, so the same set of varieties grown at multiple locations or multiple years, we can rank on the x-axis the locations or the years, the growing environments, by the amount of stress. So if we look at, in red line, the average yield of all of the entries in the experiment, and we look at the particular environment that had the lowest average yield, we can define that as a high stress environment. And then we can look at the environment that had the highest yields and call that the lowest stress environment. So we rank our environments based on average yield. Then we can look at individual, I'm sorry, not the red line, this black line here is what we were talking about for average hybrids. So we can look at the average, and by definition that's there, but we can look at other lines, individual lines, and ask how did they perform in these environments ranked by the average yield. And we can find lines like this blue line, varieties or hybrids like this blue line, which under high stress environments produce better than average, but under low stress they perform worse than average. Now, you might wonder why somebody would grow that variety, but in fact these varieties are extremely important in the developing world. So if your whole life, your family's income or food supply depends on whatever you produce on the farm, you cannot afford to have a crop disaster. So it's worthwhile to give up the potential of higher yield in a great year to protect yield in a stressful year. In the developed world, where we have things like crop insurance to protect, and people have savings, to protect for a really bad year, a grower might be willing to grow, they typically are willing to grow hybrids like this red line, which perform poorly under high stress, but in a good year, and a good year is one where they can actually control the environment to a certain extent by the application of fertilizers, pesticides, irrigation, they can benefit from higher yields. And the non-parallel nature of the red line and the blue line is in fact a genotype by environment interaction. So the red genotypes perform well in good years and worse than average in poor years, where the blue has the opposite relationship. Now, if we were able to, what we would really like is something like this green line that performs beautifully across all the environments, but I guarantee that if we produced a hybrid like this, somebody would say, well, how come it doesn't go up when we have a low stress environment? And they would like something here, which means we'd have to come back here, and then we'd have to go up anyway. But what is the genetic basis of this genotype by environment interaction? If we're going to exploit this in breeding, we need to understand how it works. So one of my graduate students has been, Aaron Kusmak, has been mining historical data. We have data for about 25 traits on thousands of genotypes grown in 20 environments. So we have the genotyping information, we have the phenotyping information, we can set up something like this, and then we can look for associations between variants in the genome, SNPs, and two things. One is the intercept on the slope. So we've got all these varieties. We've got 5,000 different inbreds. And so we have 5,000 lines. And so for each line, for each trait, there's an intercept. And we can find genetic variation that is associated with differences in the intercept. And that's the trait value per se. But we can also use as a value that we're trying to find associations in the genetic code with the slope. So just as the blue line and the red line have different slopes, we can identify SNPs associated with slope, which is the G by E term. It's plasticity, actually, more formally. And so he did this experiment, and he found lots of associations, both with the intercept and plasticity, or the slope. And the very important finding from this was not the specific genes, but that the genes that control or are associated with intercept, the trait per se, and the genes that are controlled or associated with slope are different. So this supports the structural gene model, which was proposed back in 1989, but there's been very little data to test this versus the alternative hypothesis, that different sets of genes control traits and stability of those traits. And this is very good news, because it means breeders can essentially aim for something like the green line. I don't think we're actually going to achieve that, but we can get high yield and stability with appropriate breeding practices, is what this says. The challenge is, if we look at the genes that Aaron found associated with slope and intercept, or anybody who's doing GWAS studies, genome-wide association studies, we find genes associated with agronomically important traits in maize. Most of those genes, there are no publications at all about those genes, none. And for a very small subset, there's one publication, and then beyond that, most genes, there's nothing more. So this means it's quite difficult to figure out what these genes that are associated with important agronomic traits are actually doing, and hence it's hard and difficult to come up with the next set of experiments to test these relationships or to use that information in a breeding program. This is a slide I borrowed from Carolyn Lawrence Dill, who's here in the audience. She and her graduate student have looked at the Go term annotations. So Go terms, gene ontology terms, are, even if we haven't conducted, if we don't have the full function of a gene, sometimes with Go terms we have some clues about what it does. The bad news here, Gokul's work has shown that essentially all of the Go terms that are available for maize were automatically assigned via similarity of the gene to genes in other species, which themselves are often assigned automatically by relationships to other species. And of course, our confidence in these automatically assigned Go terms is relatively low. So that's bad news. We need to develop a greater understanding of the functions of genes that, in fact, control and contribute to agronomically important traits. So let's move on to the next bullet, which is to develop new phenotyping technologies, sensors, robots, UAVs, and new methods for analyzing the resulting data. So in my own group, as we thought about building automated phenotyping systems, we recognized that we want to phenotype plants at multiple time points during development. We're not simply interested in the final height or the final yield of a plant. We want to be able to study dynamic traits, such as the rate of development and the plant's response to the environment. That means we need to be able to measure plants multiple times during the growing season. And we want to be able to phenotype entries in multiple environments to study the effect of E and G by E. That means we need to be able to be in a technology that can be deployed at multiple sites. And for maize, at least, much, not all, but much of the exciting action is only visible from the side of the plant. So using a UAV to look down on a plant, you can't see, for example, the ear, which is where the grain is. So that is another design consideration. There are all kinds of interesting technologies out there for phenotyping in field situations. There are these gantry systems. This one happens to be in Germany, where you have a fixed gantry and you can put various sensors and cameras that can move back and forth over the field. This won't work for our needs because we want to be able to, this would allow us to measure plants multiple times during the growing season and at extraordinary resolution sometimes. But we can't deploy these gantries at hundreds or dozens even of sites around the country. They're too expensive and they're certainly not mobile. We can use UAVs, and I'll talk about that a little bit. We can build autonomous robots that go through the field collecting data. So these can be useful to us. This doesn't let us look at the side. This has some constraints. We can't measure very often, maybe only a few times a week. And we can't move these from field site to field site unless we can buy lots of them, I suppose. But they're big. We can't move them around. This is another system, a rail system from Ikersat in India, where, again, they have a boom that moves back and forth over pots. Again, not suitable for us looking at multiple locations. So what I'm going to do is focus on inexpensive stationary field-based sensors, though I will talk a little bit about UAVs. So the approach that we came up with, and as I say, we're using a number of approaches, but the one I want to talk about today is time-lapse photography. This is not new. Back in 1900, and I thank Edgar Spalding for pointing this out to me after I gave a talk about this, he said, you know, back in 1900 rather, they were proposing using essentially time-lapse photography to show phases of several days, weeks, growth in a minute or so. Well, and Edgar is right. And I looked on YouTube, and from 1933, here's a nice example of time-lapse photography. There's a nice soundtrack.
The outer skin has been removed from this particular seed so that we can watch the growth of the young plant within.
We can watch the shoot.
First, the root appears for the right of way, and the weaker has to choose a different route.
But they only had one camera taking picture of one plant. What we wanted to do was make this high throughput. That was, of course, happening in a controlled environment. We wanted to make this high throughput, and we wanted to move it to the field. Now, initially, we didn't have it in the field. I was inducted into this area of investigation by James Schnable, who's at the University of Nebraska, a very nice fellow who I've known for a long, long time. And he came to me one Christmas, and he had built this thing with a Raspberry Pi that controlled a cheap cell phone camera. And what he was doing was programming this to take a picture every 5 or 10 minutes. And here he is looking at our bird feeder, and we were studying how birds respond to different grains. And sorghum, if you feed birds sorghum, which is what's in the cheap bird seed that you buy in the grocery store instead of the bird food store, the birds basically kick the sorghum off. They don't want to eat it because the breeders have carefully added to sorghum compounds which discourage birds from eating the grain in the field. Millet, on the other hand, they like that, which is one of the problems with millet, I guess. Now, building a collaboration, you absolutely have to have input from both sides, right? And so James did all this. I provided the wooden adapter that attached his Raspberry Pi and camera to my tripod. So I provided both the resources as well as this piece of wood. And that's been the way our relationship has been for many years. But this idea was clearly very exciting. Young in my lab was a postdoc a couple of years ago from Korea. He's now back there. Shrikant was a scientist in the lab. He's now in India at one of the new IITs. But what these guys did was they, with help from many others in the lab, is converted this into a technology that we could deploy in the field and do so in a very high throughput manner, which I'm going to show you in 2 slides. But first, I want to show you some data from James when he was at the Danforth Center in a greenhouse using his Raspberry Pi and his duct tape. He was looking at some plants which he had stopped watering, taking a picture every 10 minutes. And we all know what happens is if you stop watering plants, they wilt. But what had not been observed before was how that process of wilting occurred. And so we're looking over here, and I'm talking just like some folk singer who's waiting for the thing to come around on the... Anyway, why isn't this... Eventually, yeah, okay, there we go. All right, watch these leaves. Here they go. So what you're seeing is the process of wilting, which any of us who are plant biologists who've had a student fail to water the greenhouse over the weekend sees the outcome of this. But watching the process occur, I've shown this all over the world. Nobody has said, you know, I've seen this before. What's also interesting is this genotype here, which has a transgene in it, and it is wilting more slowly. Now, it will ultimately wilt. So if you look at an endpoint, this genotype is indistinguishable from the others. But by watching this go in progress, we can distinguish the phenotypes of lines that would be otherwise indistinguishable if we just looked at the endpoint. So that was enough to convince us to try this in the field. And we did it in 2015 was our first large-scale experiment. So we had solar panels out there because we didn't have, this is our UAV taking these pictures. We had poles, and on each pole we had a camera, because we upgraded from James's cell phone cameras. We got relatively inexpensive consumer-grade cameras, placed them in almost waterproof cases. We had USB cables running all over the field. That box there had a battery in it. We collected weather data. And then by taking pictures every 10 minutes, we could watch things such as the rate of growth of different genotypes. Each of these 5,000 genotypes in this field, we had 13 million SNP markers on it. So we can measure growth rate. I'm not going to show this whole video, but we can also watch anthesis, the process by which plants produce pollen. And there's enormous variation from line to line. This is an important trait for seed companies when they're making hybrid seed. They have to cross a female line by a male line, and many of the male lines have trouble producing sufficient pollen over the right duration. So we wanted to understand the genetics of that. What we were really hoping, though, was to watch the drought process occurring in the field in Ames. And to my dismay, but to the pleasure of the Iowa corn growers, we did not have a drought. In fact, we seldom have droughts in Ames. So, well, I'll get to what we did about that. But first I want to talk about the IONA data. So we're taking lots and lots of photographs, something like 700,000 per month. This is 5.5 terabytes of data. And we have all these images. I wasn't able to convince the graduate students to just sit there and look at the pictures and tell me what was going on. So we started collaborating with Bhaskar, who's here in the audience. And he taught me something very interesting. I mean, the initial response of a biologist is to say, well, let's just extract those tassels from the images. We can just ask the computer to do it. And I was naive enough at the time to think, yeah, well, let's just tell the computer to, mark out the tassels, and then we can extract some numbers from there. And one of the things Bhaskar taught me or made me aware of is that computers are actually quite good at replacing or doing what we learned in school. They are much less able to mimic what we learned before we go to school, like recognizing mommy's face versus daddy's face. But because of what we're going to be talking about over the next 2 days, it is possible with training sets to develop approaches to extract, for example, the tassels from here, or as one of Bhaskar's students did, determine the angle of these leaves so we don't have to look at picture by picture, but in fact, we can get changes in leaf angle over time. Coming back to the drought. So Ames, it rains in Ames. Again, to the pleasure of the farmers. That's why it's the center of the Corn Belt. But what we did is we moved out to Nebraska, western Nebraska and western Kansas, where, according to all reports, it's dry. And Colton McNinch, another grad student, his dad farms out here in western Kansas. And his dad told me that every 5 years, they get they get 2 years where the crop fails completely from drought, 2 years where they limp along, and there's a 5th year that pays for the other years. So with high hopes for drought, not Colton's dad, of course, but on our fields, we hoped for a drought. So we set up the same kinds of systems where we have the cameras, that's in this plot here. But we also had the same genotypes in replicated yield trials that were irrigated and yield trials that were non-irrigated. And the idea was to look for surrogate traits. We wanted to find, with the camera, were there things that we could see with the camera, behavior of the plant, that would predict the difference in yield under irrigation versus non-irrigation, drought tolerance? Or could we see something in the camera, some surrogate trait that would allow us to predict which lines performed well in the absence of irrigation and under drought stress, per se? And this is because these replicated yield trials are expensive and limiting. So if we could set up a very simple surrogate trait that we could see with the camera, this would allow us to, again, prioritize which lines go into the expensive yield trials and not waste space and money working on lines that clearly aren't going to perform. Well, guess what? It rained in in Kansas and Nebraska last year in 2016. So we didn't get drought tolerance, but we were still able to get very interesting information. So Colton had some high school students that he engaged with one of the science teachers in the town next to our plots, and they measured the height of the plants at multiple time points during the season, and then we also extracted the height from our images, and we're able to see that, in fact, the high school students did well as compared to the cameras. That was good news. That meant we could plot the growth rates. This is one particular maize line, one variety, and we had 5 plants visible to the camera. And you can see there's some variation from plant to plant, but they all had this sort of sigmoidal curve. Now, once we have this, we can extract the growth rate from this curve, and we can plot that. So here we have just 2 genotypes, one in blue and one in orange, grown at 2 locations, the circles and the triangles. So let's start with the orange genotype. We're measuring growth rate. Growing degree days is how long since the plants have been planted with temperature considered, but we get the same kind of plot even if we just do days after planting. So at this orange genotype, the shape of the curve of the growth rate is similar at both locations. It's offset by some time, but the shape of the curve is similar. In contrast with the blue genotype, we have a very different shaped curve at the first location as compared to the second location. So this is, in fact, genotype by environment interaction for growth rate, change over time trait. What we ultimately want to do, though, is be able to predict which lines are going to perform well under drought and which ones are not. And so the camera is one approach. Liang Dong, again, has been developing sensors for measuring the relative humidity right on the surface of the leaf. So he's developed what he calls plant tattoos, and these sensors go right on the leaf, and they can measure the relative humidity, and he can show that with our plants, that when we water the plants, we can see this changing from lower leaves to upper leaves as water moves up through the vascular system to the leaves. So, and there are differences in very preliminary experiments. There are genetic differences in the rate at which that occurs. So we think that by combining the cameras as well as Liang Dong's tattoos, along with the yield trials that we've been doing under irrigation and non-irrigated conditions, we may be able to get some predictive power. But of course, the roots that are providing the water that Liang Dong's sensors are measuring are important, because that's where the water comes from. Yep. With roots that have been developed, you can put those through an image analysis software, and you can begin to see enormous differences. So we have a set of 380 inbreds that Colton and his collaborators have analyzed using this method, and root volume varies enormously from small to large, all grown in the same environment, and root angle also varies a great deal. So we want to add this to the model. Can we take this collection of data, the genotypes, we have everything marked, the relative humidity, what's going on in the camera, as well as the root data, and begin to predict which lines will perform well under drought stress. So now I want to talk about something a little more philosophical, and that is that most of us who are scientists were trained to think about hypotheses. We design hypotheses, hypothesis-driven research. And I've run into some people that have so absorbed this important approach that anything else is heretical to them. And the point I would like to make today, which I suspect to this audience is not so heretical, which is that data-driven approaches can greatly add to our understanding of biology. And in fact, biology began with data-driven approaches, right? People walked around the world, they looked at things, they saw interesting patterns, and then they developed hypotheses. They didn't start with a hypothesis. The other philosophical piece here is we can do experiments in controlled environments, and the advantage of that is we control the environment. There is only one environment. So when we're looking at that equation of phenotype is driven by genotype, environment, and the interaction, there's only one environment, so there's no interaction term, so it's genotype controls phenotype in that environment. So that's quite nice, but of course ultimately we want to grow the plants outdoors in the field or the target environments. So I want to talk a little bit about how these two approaches of where we conduct the experiments are synergistic. So back in 2015, I showed you some video of our UAV, collected with our UAV. We were going over these plots with the UAV, with camera collecting images from above. And mostly for maize, as I've said, the interesting stuff is visible from the side. But we decided to look at these and see if we could find something interesting. It was truly a fishing expedition. So Ting Zhang, one of my former graduate students at China Agricultural University, who was at Ames for the summer to collect data, noticed this interesting pattern, which is that some genotypes, the seeds are planted with random orientation. And when they come up, maize has alternate leaves, and so it's almost 2D. And this is what we would expect to happen, which is that the leaves are, to use the technical term, higgledy-piggledy. But we noticed that some genotypes had this unexpected pattern of being parallel to each other. Like a crystal lattice kind of a thing. This was interesting. Maybe the plants were, because this couldn't happen by chance, the numbers that we were looking at. The seeds wouldn't all land in the ground in a way that would lay them up like that. So maybe the plants were turning in response to their neighbors and orienting themselves. Well, to ask whether or not there was a genetic control for this, Ting conducted a genome-wide association, and she found a number of hits. Most of these hits were unannotated, just like Aaron had found with his experiment. But one of them had some annotation, which may not be correct, but at least we could make a story about this annotation. And I think any scientist who can design a good experiment can come up with a good story for whatever annotation you come up with here. So that's a joke, but more seriously, we need better annotation for genes, or else we can't really design the subsequent experiments. But this data-driven approach led us to develop the hypothesis that these plants are rotating. So we set up an experiment in the greenhouse where we watched some plants. So now the cameras are looking down on the plants. We're doing time-lapse photography, and we're watching these plants. And indeed, some genotypes, but not all genotypes, do in fact go through rotation of leaves. The phyllotaxy is changing during development. So that was enough to convince us to move out into the field again and set up biparental populations derived from biparental crosses with lots of genotyping data. So we could ask, again with cameras, looking down on plants in the field, we could watch these plants and ask whether we can map the genetic determinants that allow plants to rotate or not rotate. Now, again, we had huge amounts of data, and this is a challenging problem of how to track individual leaves over time, and this is something we're working with Bhaskar's group now. It is a big challenge, but we talked a couple days ago, and he has some great ideas about how we might be able to do this, and I'm saying this publicly so that Bhaskar will deliver. All right, but even if we have data, if we can convert all of those time-lapse videos to numbers, we still need to try to figure out to make some sense of it, and this is where biologists need to come in, and so Yan-Zhou has been working on that, and I'm not going to go through details there. Except to say that there is some sense to all of this. You remember this curve, this yield curve, this increase in yields that we saw over time. It turns out work of Duvik back in 2005, looking at old varieties and new varieties, that most of this increase in yield is not due to increases in per plant grain yield. What's happened instead is since the mid-30s to now, plants have been bred to tolerate crowding better. So mostly what's happened since the mid-30s is plant densities have increased. Well, so that makes sense, right? If plants are able to recognize their neighbors, just like people on subways, they somehow manage to crowd themselves in, and that makes a lot of sense to me. What doesn't make sense to me is B73, which is the progenitor of many modern hybrids, doesn't rotate. So we're still sorting this out, but it's intriguing. This slide is basically just to show that the experiments conducted in controlled environments and target environments really are synergistic. We began with a data-driven approach in the field. We moved to a controlled environment. Then we moved to the field. We moved back to the growth chambers or greenhouses. And this summer, we're going to be back in the field. So these are where we conduct the environments are synergistic, as well as this hypothesis-driven approach is a good prelude to data-driven is a good prelude to hypothesis-driven research. So I want to finish in the last few minutes with this last point, which is building a community of plant scientists, engineers, and computational scientists. And this is something I've been wanting to do for many, many years, and well, actually since the mid-1990s. So Iowa State had hired a number of plant molecular biologists in the late 80s. And Jim Cornett, who was a mathematician, started coming over to us in the early 90s and saying that there were all these computational scientists who had amazing tools, and they could help us analyze our data. And we were like, you know, Jim was a nice guy, so we were very polite about this, but Jim, the band is on the gel or there's no band on the gel. I don't need any math or statistics to sort that out. By the mid-90s, when sequencing started exploding, it became clear we really did need help. And so Jim, fortunately, had persisted and had not given up and came back again, and he established a bioinformatics seminar that met once a week. And he invited biologists as well as computational scientists. And in alternate weeks, the biologists presented their challenges, what they were trying to do but couldn't do, and computational scientists presented solutions. And for me, this was one of the most exciting things I have ever done professionally, to listen to computational scientists talk about algorithms which reduce the search space without affecting the final results at all, thereby speeding it up. I mean, these were beautiful algorithms. And I was gratified to see in the New York Times recently that there are studies that the same part of the brain that lights up when you listen to beautiful music will light up when you look at a beautiful, well, when some people anyway, look at a beautiful algorithm. So that really was, that was amazing for me. And we have since deployed this Cornett model for building a predictive phenomics community. So we began by inviting not only the plant biologists and the computational scientists, but also the engineers. It was clear we needed engineers. And I have to say, working with engineers is extremely gratifying because they like hard problems. They consider them gifts. Maybe not too hard, but they like reasonably hard problems, and they consider it a gift, and they are very driven to solve those problems. Where if I come to a plant biologist with a problem, they listen sympathetically and say, good luck, you know. So we met for a couple hours every Friday during the fall of 2014. I provided lunch. You know, the joke is that if you provide pizza, you can get graduate students to do anything. If you have, with faculty, you need to upgrade to, you know, gourmet sandwiches. But they did come. I had a lot of fun with this. We had internal speakers, 1-2 a week, with lots of time for discussion. About 30 faculty came most weeks. I mean, the same 30 came most weeks for the whole semester. People really enjoyed it. I enjoyed it. It was a chance. There were no graduate students or postdocs. It was just faculty. People could ask the dumb questions that you need to ask at the beginning of cross-disciplinary collaborations. It was a lot of fun. And it began to pull together people from these different disciplines who wanted to work together, who could see that they had interests and skills, interests that were similar and skills that were truly complementary, and there was some personal chemistry. But I was able to go a little further because I had been given some resources by the university when I was appointed director of the Plant Sciences Institute. I had done my postdoc at the Max Planck Institute in Cologne, and the Max Planck has this model, which is also the same one the Howard Hughes Medical Institute investigator model, which is not to invest in specific projects, but to identify really talented, driven people, give them resources, and let them have at it. So our approach was to invest in people, not projects. We went through a selection procedure that involved off-campus reviews of individuals, very distinguished reviewers, and selected 16 PSI scholars. Biologists were about half of them, and then the other half were computational scientists and engineers to help us build this community. And we provided these scholars with stable, multi-year funding, and they could use it for anything they wanted to, as long as my administrative assistant said it was legal. So I want to give you just a few snapshots of how this is a progress report. This is an experience that we are not at all, by any means, done with, but the results are very promising. So these are the PSI scholars here. These lines show collaborations in the area of predictive phenomics. And most of these people were not working in this area when we began this in 2015. So these are essentially all new collaborations. So we've got this network of community. When I go to upper administration and talk about this experiment, as they called it, we have some metrics. I don't think these are necessarily the right metrics, but they certainly are promising. The group has brought in 71 grants with $15 million. That's non-redundant counting. 100 in just the first 2 years, and its curve is going like this, I believe. 136 publications, and this does have redundant counting because they're all collaborating together, so they're co-authoring papers, and then 139 speaking engagements by the 16 PSI scholars. That's good, but they're also reaching out to the rest of the campus. So again, PSI scholars now in white, non-PSI scholar faculty just at Iowa State in the black circles, and you can see that particularly some of these PSI scholars are building they're really hubs in this network of researchers, so that what's going on within the group of PSI scholars is also greatly influencing others on campus. And we've been able to secure a $3 million training grant from the National Science Foundation to cross-train the next generation of students at the intersection of engineering, plant science, and data science to address the key challenges of our century. So to summarize, I believe data-driven approaches and modeling are going to facilitate the development of new crop varieties that exhibit yield stability, even though we have increasingly variable weather patterns. And this is not trivial. Our agricultural systems were developed over the last 10,000 years during a period when weather patterns were very stable looking geologically. So we have an enormous challenge ahead of us. And to meet that challenge, we've got to have collaborative teams of scientists from multiple disciplines, and they need to be truly collaborative teams where people trust each other and are comfortable asking the simple questions to gain enough understanding so that they can actually work together. We need further investments in the developments of different kinds of sensors, robots, and certainly algorithms and other analysis methods. We need support for data collection from coordinated multi-year networks of field sites along the lines of genomes to fields. And we need functional analysis of genes involved in agronomically relevant traits. And this multidisciplinary environment that gets created by pulling together these teams is an amazing place to train graduate students in STEM, and it also has the ability to stimulate the ag-related startup ecosystem and promote economic development. So with that, I will answer any questions that you might have. Thank you. Questions? We've arranged, I think the hub's paying for the questions, right? They're free to everybody in the room here. Free questions today for the next 5 minutes.
Do the answers cost?
Yeah, that's good. See, Joe's an economist. Yes.
I have a biology question.
Yep.
So about the plants that rotate.
Yeah.
Are they orientating themselves to get more sunlight?
This is a great question. We are trying to set up experiments to do this. Last summer, the position of the embryo in the seed determines where the orientation of the first leaf or two. So we put them all in the same orientation. The short answer is I don't know, but I have lots of experiments in mind, some of which are underway, to try to answer questions like sunlight, proximity to neighbors. I don't know what else. We're going to try and figure it out. There are different ways it can rotate. It can be rotating by where the leaf comes out of the shoot-apical meristem. It can also be differential growth on the side of the leaf. There's a lot of stuff to do still, so we're working hard on that. Other questions? Back there, yes, please.
So the yield improvement since 1930s that you presented, how much of that is per plant productivity versus the seed density per unit area?
Yeah, so the question is, the yield increase since the 1930s, what is the proportion of improved agronomic practices, fertilizer, for example, the comment here in the front row at the beginning of the talk, versus improved genetics? And estimates vary. When plant breeders do the study, they're about 60% genetics, 40% agronomy. When agronomists do the studies, it's flipped the other way around. But let's say about half and half is improved agronomic practices and improved genetics. But it's important to recognize that the corn of today is not the corn of 1930, and so the improved agronomic practices that we use now wouldn't have the same effect on the corn of the 1930s either. These two disciplines have evolved in parallel. Other questions? Yes.
So it seems to me that there's an interest from the scientific community on interpretation, being able to interpret
Right.
the outcome of the modeling
Right.
that you're doing. And the push from the machine learning community is very much towards
Black box.
deep learning, black box,
Yeah.
Get better prediction,
Right.
but not so much concerned about why.
Right.
So could you talk about that?
Yeah, this is an interesting tension, and it is also the tension between a geneticist and a plant breeder. So a plant breeder is charged with developing a new variety, and how you get there matters less than the details of which genes and alleles have been selected. Where the geneticist really wants to understand the nature of genetic variation and how you compile this into interesting packages and produce something that's different. I was reading an article last night about some young person who's developed corn which grows, I guess it was 44 feet tall, I think, by introducing both interesting mutations as well as controlling the environment. He was clearly not aiming for the production market. So both are important. I would say, if we can come up with predictions that are black box but accurate, the farmers will love it, the breeders will use it, absolutely. But I also think, as a scientist, I also want to understand how all that's working, and I recognize, sadly, that the machine learners may not be able to give me that, but if I have really accurate predictions, then I can also go in and start looking at groups of things that are this and that, and I can look at the DNA and try to figure out what's going on. Other questions? Joe?
Any idea why B73 does not have that?
No, that surprised me, but yeah, B73 is not rotating. We've got a lot more work to do on this. These are early days. You could argue that my analogy of the crystal lattice is all wrong, that you want plants that are higgly-piggly, that you'd get better light capture under that, but I don't know. Yeah, there was another. Yes?
Back to the orientation question.
Yeah.
What percentage of the varieties that you looked at are
Rotators?
versus
It is a minority. It's not a tiny minority, though, and I unfortunately can't tell you exactly the numbers, but, double-digit percentages, but less than 50. There was another one somewhere? Oh, yeah.
Just wondered, I saw you're using UAVs for visual inspection of
Yes.
crops. Are you planning on, or do you have any interest in using them for things like digital phenotyping of crops?
Yeah, so there are lots of people doing that, and I know there are some real experts here and also further east, some of who are in the audience to do this. Those are most useful when you're looking at plots or fields, and we're mostly looking at individual plants. And so that approach is less useful to us. Yeah, so I can't do everything. Not that it's not important. Anything else? Oh, yes. Nick.
So you mentioned about the poor annotation of the genome, but also talked a lot about predicting phenotypes. So where does genomic prediction fit between these two, in which you want to predict phenotype, but you kind of don't care about annotation?
Yeah, so genomic prediction, for those of you that aren't biologists, is an approach to look at variation, all the SNPs in a genome, use that information to predict the performance of a particular individual based on its genotype. And you don't need to know anything about gene function to do that. Genomic prediction is a very powerful tool. It came out of the animal community, so this is real synergy between the quantitative geneticists, particularly in the dairy industry, and the crop world. It is transforming breeding, and it's very much a black box. So this is a good response, which is we have black boxes that are being used in breeding, and they work great. I would argue that ultimately we do need to understand it because there are limits to what we can do with black box predictions, at least with genomic selection. Yeah. Yes?
So this question may be a little bit further from your main interest that I wanted to ask you. Are you familiar with the Global Agricultural Monitoring, which 82 countries have signed for monitoring yields of crop, rice, wheat?
No. You're right. It's outside of my area, but yes, sounds like a great idea.
So, there they use remote sensing imagery, which is
Yep.
actually pretty coarse. You can't tell
Right.
things about individual plants, but
Right.
you can tell about the overall.
Right.
And it's now being used by policymakers and companies to, your fourth vision
Right.
to anticipate crop failures and take actions. And the main question was, what's the trade-off between looking at individual plants with vision and trying to measure angles and all these little details
Yep.
versus doing remote sensing worldwide?
Right, so the question was the comment initially was pointing out that there are 82 countries, I think you said, that have signed an agreement to do remote sensing for agriculture, and that this is lower resolution, but still very useful for predicting crop yields and anticipating problems such as famines. And contrasting that with the high resolution, but much lower throughput approaches that I've described here. And I would see the differences for breeding and genetics. We need the high resolution. We need to look at individual plants with different genotypes. For predicting agricultural production, sometimes looking above coarse resolution is absolutely adequate, and we can look at large areas. But I think also these two can be synergistic in the sense that we can look at high resolution at small numbers, compare those to, say, satellite images of those same small numbers, and calibrate and learn something about why there are patterns that we can see with satellites by comparing to the high resolution. Yeah. All right, thank you very much, everybody.